Morning.
Good morning. Manasi, invite all to join.
Yes, sir, I'll do that.
Okay, students, welcome back. So, another session. Let's wait for 2 minutes. So, all will join, then we'll start. Okay, students. So now I think most of you are here, so we can start. Okay, so we were at module number 1, and we have talked about we were talking about your enzymes, and in that we have done with the restriction enzymes, your nucleases, what they are, what different types of nucleases are, and the most important one is your restriction endonucleases, how they're being discovered, and what's their utility, and the recognition site, their types, the enzymatic classification also bit we talked about, and what are the requirement, different types have different requirements, and some other class of the typical 3 restriction endonuclease. We have some more classes, that's the type 4 and type 5, and also the type 6 that we call the artificial restriction enzyme. Then we do the some comparison, but they are, so as in the first we mentioned comparison on the basis of their requirement, their composition, their cleavage site, etc. So all things are here. So similarly, this is the second table, just few things are here and there, most are the common. And how these enzymes are going to work, their mode of action, and after the action what they produce, what type of cuts they do, blunt and sticky, and what are the utility of their blunts and sticky ends, different enzymes, important enzymes, how they named, and what are the recognition site, and after recognition whether they produce the sticky end or the blunt end, then how you name them. So in the naming them depend upon their source, genus and the species. And the star activity is if your enzyme start behaving abruptly, what are the reason behind that? So that is the star activity. And then we talk about the isomerism in your enzymes, restriction enzymes. 3 different types of the isoforms are there: isocytomer, neocytomer, isocodamers. Then application done, and after that we talked about the ligases also. These also we have done. So the job of ligase is reverse to the job of your restriction enzyme. So they are breaking the phosphodiester bond, they are generating the phosphodiester bond, and what the requirement and what the mechanism of action. So three things are there. First activation, then loading over your enzyme, then joining, and the energy is being required in this process. So energy is either provided by the NAD or ATP. Then the utility, then the recombinant technology, we utilize it for joining two. So wherever you need to join two segments of your DNA, there comes the role of ligases. And naturally in your DNA replication, your own life, it's a very important role because it's play a role in your DNA replication and the damage repair. And after that, we were now at the third enzyme of our importance, that is your DNA polymerases. So before moving to DNA polymerases, I would like to ask you, any one of you have any query regarding what we have discussed yet? If you have any query, then please raise it now. Otherwise, then we'll move further. Anyone of you have any query?
No, excuse me, sir.
Yes.
Sir, what's the difference between gap and nicks?
Okay, what is the difference between gap and nick? Gap, what do you mean by the gap? Just literally meaning. I'm just asking
Sir,
literal
some missing base pair.
Yes, that is a gap. If the base pair is missing, then you will say it's a gap. And nick is just a break.
Sir,
Okay.
breakage of phosphodiester bond.
Like this is your pencil. Are you able to see? If just I break this pencil, then that is a nick. Two pieces. And then what I do, I put one break, second break, three pieces I made, and I remove one piece from in between. Then that is a gap. Okay.
Okay, sir.
So
Thank you,
just
sir.
a moment, I will show you a moment. Just a moment. Okay, so like we talk about the sequence like A, T, G, C. Okay, so double stranded DNA is there. So same thing going to be like they are complementary. So if you break from this point only in the one strand, then this is a nick. Okay, nick means only in one strand you are breaking. If you are breaking here also and you remove the T from in between, then there is a gap. Okay, got the point?
Yes, sir. Yes.
This is the difference between the nick and the gap.
Okay, sir.
Rest all things done?
Yes, sir.
Okay, so now we are moving further. So now the third type of enzyme that we are talking about. So whatever the enzyme we are talking here, all the enzymes, they are going to have role in your replication or somewhere else. So all these things are going to the third enzyme also, it is an important enzyme that is playing a critical in your DNA replication. Why I'm saying DNA replication, because as as we are talking about the DNA. So DNA means whatever in your life form, whether it is going to damage, it is going to repair, or your normal DNA replication. Whatever enzyme you are doing inside your cell, we are taking it out and we are utilizing it for our purpose. That is the thing. So now the next enzyme, DNA polymerase, as its name is there. This means this is the same enzyme that is going to play a role in the polymerization or in the DNA replication. And when we talk about the DNA replication, it simply means, if you simply, when you define, when you talk about a semi-conservative. What do you mean by semi-conservative? What do you mean by semi-conservative DNA replication? What do you mean by the semi-conservative DNA replication?
Sir, one strand it remain as it is and the other replicates.
Yes, rightly said. So the semi-conservative DNA replication, it means one strand is going to be original one. So it means As in your DNA replication, as your DNA replication is semi-conservative in nature, this means it requires one's pre-existing strand. This is again the same thing here also when you're talking about the DNA polymerase, how it is going to act. So if you want to work with your DNA polymerase, if you want to make your DNA polymerase to work, then you need your restriction. Sorry, just a moment. I'm coming back. Hello. Okay, so we are talking about the DNA replication. So it means the first thing, DNA polymerase required a template strand. If the template strand is not going to be there, it can't synthesize your DNA. So the polymerase means that the polymerization. So it have to polymerize, means your nucleotides are there, like A, T, G, C, these four nucleotides are there. The role of this DNA polymerase is the formation of a phosphodiester bond between two nucleotides. As you will say that the DNA ligase is also doing the same job. But the main thing DNA ligase cannot incorporate your new nucleotide. Whatever the nucleotide is there, it can join that only, whatever the present. So it can join two strands, but it can't incorporate new nucleotide inside it. Okay, so that job is done by the polymerases. So polymerase can add the new nucleotides. Okay, so when it can add the nucleotide, it can add only the nucleotide when you are having pre-existing strands, or that it can do the process of replication. Now, if any one of you tell me how the replication occur, how the DNA replication occur. So how the DNA replication occur?
Sir, so can I give just a brief?
Yes.
So, the double-stranded DNA will separate at one particular point, and it forms a particular replication loop.
Yes.
And
Manasya,
then,
stop. Just stop a moment. So continuing
okay.
in between, I will say something. So as Manaswini mentioned that in the replication, the first process is unwinding. Means your double-stranded DNA is there; it must form the single-stranded form. So this is the prerequisite here also. If you want to work your DNA polymerase, then your DNA must be in single-stranded form. Yes, Manaswini, please continue.
Yes, sir. So once the replication loop is sort of opened, then there are again several enzymes which ensure that the loop stays open and the stability of the remaining DNA is still maintained. And then after that, DNA polymerase will bind to one particular site on one of the single-stranded DNA, and it'll start adding complementary nucleotide base.
Manaswini, something you are missing. You are missing something.
Um, okay, sir, I'll have to think because we learned it long
Yes, sir.
ago.
Yes, rethink it, and you are missing something that is very important thing. You are missing a role of a primer.
Oh, right, right. So, so basically, primers are small sequences of nucleotide bases, which are, as since DNA polymerase can synthesize only 5 prime to 3 prime. In the beginning, the primers have to be attached, and then from there, it's like a starting point, and from there, the DNA polymerase is able to add the complementary base pairs.
Yes, as your
And
Manaswini was saying that you need a primer. So DNA polymerase cannot synthesize a new strand. First thing, what DNA polymerase can do? DNA polymerase only can extend. What it can do? It can just extend if the pre-existing strand is there and it is having a remaining single stranded portion, then it can extend that sequence. To that, Manashin mentioned that it is a primer. So you need a primer. If primer is there, if a short stretch of DNA is there, and rest of the portion, if it is a single stranded, then your DNA polymerase can extend that. Extend it, it can add new nucleotide there. If there is no pre-existing strand, if you are having only a single stranded DNA, DNA polymerase is not going to work. Okay. So that is the one. Now, Manishni, please continue.
So DNA polymerase, it starts, adding the whatever needed base pairs, and after that, eventually it'll
At
reach
which end it add? At which end it add?
it only adds from 5 prime to 3 prime.
Yes, and only from 5 prime to 3 prime. It means it is adding new nucleotide at 3 prime end. Okay, and at the 3 prime end, who is present? Who is present at the 3 prime end? At the 3 prime end, there is a OH group. So
Hydroxyl.
that you are calling 3 prime OH. So this is also a prerequisite of the DNA polymerase, is that at the 3 prime there must be an OH. There must be a hydroxyl group at the 3 prime. If the hydroxyl group is missing at the 3 prime, then your DNA polymerase won't work. Okay, so these things that we are discussing now will also use in the future, also for some future enzymes that we are talking about. So this is again the prerequisite. If you need a 3 prime OH uh, at the 3 prime end of your primer. Okay, or even whatever the existing strand, half existing strand is there, simply as replication fork Manaswini mentioned. So that type of things are there. Okay, so continue Manaswini, something else you want to say?
So just that eventually it will reach a particular termination sequence or a stop point, and at that point it will stop replicating. But it also will, while it's replicating, it will also proofread the whatever strands it's adding to ensure that there are whatever base pairs it's adding to ensure that there are no mistakes that happen during replication. So that is the gist.
Yes, well said, Manaswini. So whatever the Manaswini mentioned about the DNA replication, that things are connected with the DNA polymerase, the action of DNA polymerase. So we need all those things as we are talking about in your DNA replication here also. So as the DNA replication is semi-conservative, so DNA polymerase can work in a semi-conservative way only. DNA polymerase enzymes are specialized for different functions. So there are three functions are there in it. It's not just one function that are present on your DNA polymerase. What are those functions? Those functions are two types of things. One is the nuclease activity, and second is the polymerase activity. So two types of the activity are there. One is the nuclease. So nuclease activity, but what do you mean the nuclease activity? What is the nuclease activity? What are the nucleases? What are the nucleases?
So they cleave the phosphodiester bonds of
They cleave the phosphodiester. So now here we are talking about the polymerase. The role of the polymerase role is adding the nucleotide. But one more property these nucleases, these polymerases are having, that is the nuclease activity. And nuclease activity, we have two types of it, is the exonuclease activity and the endonuclease. So what are the difference between the exo and endonuclease activity? What are the difference between exo and endo? Exonuclease and the endonuclease activity?
Sir, it cleaves at the end of the
Yes,
DNA.
exo cleaves at the end and endo.
Uh,
Endo cleaves
between
within.
the DNA.
Yes, so the polymerase that we are having, the DNA polymerases, they are having the exonuclease activity. So exo means from the ends, they are not cutting within. So where their ends are, they found the ends, so they can remove the nucleotide from the ends. So now, from which end they are going to remove the nucleotides? They are going to remove the nucleotides from the replication site, because DNA polymerases are present at the replication site. So if they found ends at the replication site, they can remove it. So what type of the exonuclease activity they are going to have? One is the 3 prime to 5 prime. So replication is running from 5 prime to the 3 prime. Okay, so this is a reverse activity. This means in the back direction it can remove the it can remove the nucleotides. Second is the 5 prime 3 prime, means in the direction of your replication it can remove the nucleotides. So two types of the activities, three types of the activity is having. One, the polymerase activity, that is going to be the 5 prime to the 3 prime. Second is going to have 3 prime to 5 prime exo nucleus activity, that means the reverse direction, and 1 5 prime to 3 prime, that means the forward direction. So how these activities are going to work? One thing is the in the reverse direction. So as Manushree mentioned that it have the proofreading activity. Proofreading means seeing backward, checking. When you write it, then you have to check it, means you have to read it again. So this simply means you have to move in your backward direction. So this is the 3 prime to the 5 prime nucleus activity that we are talking about. It is a proofreading activity, means your nucleus is moving back and reading whether it have inserted a right nucleotide or not. And if it found that it have inserted a wrong nucleotide, then it can remove it. Okay, so this activity is from the proofreading activity, that is the 3 prime to the 5 prime. Then coming to the 5 prime 3 prime exonuclease, for what it is going to be? This activity is to remove the pre-existing strand. Like if you see here, like we are talking about A, then T, then G, C, A, T, G, and C. Now the complementary is going to be G, C, A, and G, C, A, and T. So these are joined, 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 joined. Okay, so now this is the 5 prime. This is the 3 prime. One, this is the 3 prime. This is going to the 5 prime. Okay, so the first thing that's I mentioned, polymerase activity. Polymerase means now from here it have to extend in this direction. This is going to be the 3 prime. This is going to be the 5 prime. Okay, so now polymerase is going to come here. It have the single stranded portion. Now it can insert a nucleotide here, that is a T, and will join it. So this is a polymerization. Now the next thing is there. Now these strands are going to be there. Either what you need, your nucleus must leave the place and ask ligase to come and join this strand. Or what it can do, it can remove these nucleotides which are coming in between. So if it remove these nucleotides, then what is going to do? Then it extend the new strand. It will make a whole new strand anew. So this activity is called the 5 prime 3 prime exo nucleus activity. Okay. So these three types of the activities are present in your DNA polymerase, and depending upon the R requirement, we utilize these activities. As you know, if you see, if you are working in the laboratory, if your experiments are performing, in that case, we don't want this activity, this exonuclease activity. Somewhere we don't want this or this also the activity. We just want only the polymerase activity. Or if you want both the polymerase and the proofreading activity. So depending upon our requirement, we can remove or we can add the activity into the required enzyme. Okay, so this is the just mechanism of action, how these polymerases are going to work. As I mentioned, you get the 5 prime 3 prime direction they are going to move. This is the pre-template strand. So over this template strand it can extend. At the 3 prime there must be an OH group, and this OH group ATP is going to come. This ATP is going to, sorry, now it is the ACP. So this ACP, okay, sorry, CTP. This CTP is going to come, and this CTP is going to pair with a G, and then the sugar is there. Sugar is going to have a 3-phosphate group. This phosphate present at the 5 prime is going to make the phosphodiester bond with the 3 prime OH. So this process is done by the DNA polymerase. This action is called the polymerization. So this is all the same thing that we are talking about in the DNA replication. So nothing is new here also. Whatever we are talking about DNA polymerization, same thing we are applying here also. So each DNTP is going to provide the nucleophile. Nucleophile means this 3 prime OH you need, and for the next round of the PPA pyrophosphate. Nothing. Okay, so now this is the new nucleophile is going to be available. Then the reaction is going to be continued. Okay, so this is the same thing that we are talking about. So how it is going to work? This the primer is there, so ATG. Then it have to extend. So newly synthesized strand it will extend. CAT is the new strand it will synthesize. So this is the action of your DNA polymerase. So DNA polymerase require a template and it require a primer to extend. If the template and the primers are there, then only it will do the work. Otherwise, it won't work. So then from where the DNA polymerase are going to come? So as we all talking about since the first slide, we are talking about whatever the enzyme we are talking, all the enzymes we got from the bacteriums, your prokaryotes. So here also the polymerase we are going to use, we are going to work. They are going to bacterial in origins. So if you remember your molecular biology classes, there are the bacteria have the three different types of the polymerase: polymerase 1, polymerase 2, polymerase 3. So different polymerases are there, and different polymerases have different types of the activity. So that you can see here, structure in pole A, pole B, pole C, and among all these polymerase, three polymerases, we require the most important that we for us is going to be DNA polymerase 1, and this DNA polymerase 1 is the polymerase that we are going to utilize for our experiments. Okay, so what are its characteristics? It have the size, you can say around 1 lakh. So 103 kilodalton is the size of it, and it is having the activity proofreading. Yes, it have others. Your 2 also have the proofreading activity, 3 also have the proofreading activity, but the 1 is going to have one more type of activity, that is the Exo Nucleus Activity 5' to 3', that is missing in the pole 2 and pole 3. So the most important enzyme if you are working in the molecular biology is going to be the DNA polymerase 3. Okay, and the polymerization rate, if you see, so polymerization rate is 16 to 20 nucleotide per second, 40 nucleotide per second, 250 to 1000 nucleotide per second. So that means if you are working with the DNA polymerase 3, and if you want to do polymerization, if you want to extend your nucleotide strand that is of around 1000 bases long, your DNA polymerase 3 is going to do in just a fraction, just a second. So you need such type of enzyme. So the enzyme we are going to use in future is going to be your Dna Polymerase 3. Okay, so this is the one. This is again the summary thing that we're talking about. So as you have seen, that we're talking about is the gap is there. Then you can fill it and utilizing the enzyme. So in this process, you are having the exonuclease activity. It can remove the whatever the pre-existing strand it was there. Okay, so these are the enzymes that we are talking about of prokaryotic in nature, but eukaryotic enzymes are also there. Depending upon your utility, if you found that you want to work on the eukaryotic enzymes, so there are the different alpha, beta, gamma, delta. In case of bacteria, you are talking about the pole 1, pole 2, pole 3. But we have the larger numbers. So we are working in that case alpha, beta, gamma, delta, epsilon, zeta, neta, theta, iota, kappa, lambda, mu, and the sigma. So these are different enzymes. These are present in the prokaryotes, so eukaryotes. Simply in human also we are having these alpha, beta, gamma. Now coming to further things. So as we mentioned in our first slide that we're talking about, the DNA polymerase 1 is going to have 3 different types of activity. So then these 3 activities, then how whether we can separate them or not. So to analyze the DNA polymerase enzymatically fragmented it. So what we did, we treat this DNA polymerase with the proteases. So which protease we utilize here? We utilize here subtilisin. So you know the pepsin, chymotrypsin, these are the different proteases. So subtilisin is also a protease. What it do? It cleaves your protein depending upon its site of action. So when you treat your DNA polymerase 1 with your subtilisin, what is going to happen? It is going to fragment into 2 portions. What are those 2 portions? First is a large protein. So that large protein is called as a clean-off fragment, named after the discoverer. So who is working on this? Who is analyzing? So the clean-off was analyzing. So on that name, that's the fragment called clean-off fragment. So what is the property of this clean-off fragment? So the property of this clean-off fragment is 3' polymerase activity, first thing. Second is 3' to the 5' exonuclease activity. So that means this is the proofreading activity and the polymerase activity. So the not required activity that was present in it was the your Exo Nucleus, that is the 5' to the 3'. So this activity was removed when you digest your DNA polymerase 1 with subtle acid. Okay, so this fragment which is having the 3' to the 5' exonuclease activity is called as the exoclino fragment. So why we are calling exoclino? Means the clino fragment are the fragments which are fragmented by the clinos by the treatment of subtle acid. Okay, so this is called the exoclino fragment. So two fragments are there. One fragment having the polymerase activity and the 3' 5' exonuclease activity. That we call the clino fragment. It is a larger fragment, and another smaller fragment that have the 5' to 3' activity. 5' to 3' exonuclease activity, and that does not have the polymerase activity and the exonucleic activity to 3 to the 5' that are called the exoclino fragments. So now if you ask what is its utility, so now we know that this enzyme have only the polymerase activity, so we can utilize it for our laboratory purpose. So we are not using whole DNA polymerase, we are using clino fragment for our requirement. Okay, what its utility is there? Synthesis of double-stranded DNA from single-stranded template. If you are having the template, if you want to single-stranded one, if you want to make it double-stranded, we will mix the clino fragment, we will mix the your ATPs, we will mix the primer, and you are going to get the double-stranded DNA. Filling the gaps, if you are having some overhang or blunt ends, like if you want to generate blunt ends, simply you know that your restriction enzyme which are like EcoR1, they are giving the overhangs. If you want to make it, if you want to remove these 5' overhang, so what you can do is simply you can add the your clino fragment. It will seal it. It will make it blunt. Then the digesting away the protruding 3' overhang. So this you can do preparation of radioactive DNA probes. So if you want to make a DNA probe, so probes are again at single-stranded DNA, and you want radio labeled nucleotide in that, so that you can do utilizing your clino fragment. So what you can do, you can you can take it single-stranded DNA, add your clino fragment DNA polymerase, then add your nucleotides. It will make a double-stranded one, and this the second strand you are going to make is going to have your radioactively labeled nucleotides. And DNA poly PCR that we are talking about now, in earlier time before thermostable enzyme that we are having, when the Kary-Mullis was describing it, at that time clino fragment were used in amplification of your DNA, but now we are using PCR because we are having the thermostable enzymes. Okay, so anything, any one of you would like to ask anything till now? Nothing? Okay, so this is again some summary of your clino fragment. So the size of it is a 329 nucleotide acid, 323 amino acid long, and having the activity. So now the next thing, the important thing is that the utility of these DNA polymerase, as you mentioned, we are utilizing these DNA polymerase in your PCR, polymerase chain reaction, and the polymerase chain reaction required a high temperature. So most of the enzymes that we are knowing that all these enzymes, they work at a physiological temperature also. That is a temperature of 37 degrees Celsius is going to be their maximum temperature. And if the temperature maximum is so much, and at that temperature if your enzyme got disabled, then how you will amplify? So for that we need to search an enzyme which is going to be not only just stable at that temperature, but also functional at that temperature. So for that type of enzyme being isolated from the bacterium named Thermophilus aquaticus, and this bacterium is a bacterium which is present in hot springs, in hydrothermal vents, and the enzyme isolated from that, the Taq polymerase, was identified as enzyme that able to withstand protein denaturing condition that is very high temperature that are required for your PCR. So that enzyme that is able to function at that high temperature Now that type of enzyme, that type of the polymerases which are functional at high temperature, if you'll ask what type of these polymerases are, these are also the same DNA polymerase 3 or that is having the same that DNA, whatever that polymerase activity that is present in your E. coli, but the only difference between the thermostable and your normal enzyme is their enzymes, their amino acid sequence. So those amino acid sequences that are present in those enzymes that make them stable to that temperature. But by nature and by activity, all the enzymes are same. Or no, if they're not the same, they are similar. Similar means they are going to have all these three type of activity. One, that is the polymerase activity. Second, the exo nucleus 3' to the 5'. Second, the third exo nucleus 5' to 3'. If you are with the polymerase 1 class, if a poly 3 class, then it is going to have only two types of activity. That is the polymerase and 3' to the 5' exo nucleus. Okay, so here we are talking about the thermostable, and the first thermostable enzyme that was described is the TEC, that is the Thermophilus aquaticus, DNA poly 3. So it has optimum temperature, that is 75 to 80 degrees Celsius. Hence, if you give the temperature between 75 to 80 degrees Celsius, it is going to work very well. It is optimum temperature is there, and it also have stability up to 95 degrees Celsius or 97 degrees Celsius. It means if you heat this enzyme for 92 degrees Celsius, 92.5 degrees Celsius, this enzyme is going to be stable up to 2 hours. And if you heat at 95 degrees Celsius, it is going to remain stable for 40 minutes. If you heat at the 97.5, then it is going to remain stable up to 9 minutes. So it can withstand a very high temperature for a quite long time. Then talking about the replication time, its speed is also very high. It can replicate 1000 base pairs in less than 10 seconds. So per second it is adding 100 bases. So simply in this way you can calculate if you want to add 1000 base pair, just give 10-second time and it will amplify your whole DNA strand. If you want to go for 2000, then you need to give a 20-second time. And at a temperature of 75 to 80 degrees Celsius, its optimal temperature is 150 nucleotide per second. Okay, so different temperature, different speed is there. So optimal speed temperature is between 75 to 80. So in your DNA replication process, we keep that temperature. So any deviation in optimal temperature inhibit the extension rate of your enzyme. So that's why we function in that temperature range only. A single TEC synthesis about 60 nucleotide per second at 70 degrees Celsius, 24 nucleotide at 55 degrees Celsius. That simply you can see that how much temperature you are going to change. So what temperature you should use while you are working with these thermostable enzymes so that they can function optimally. At temperature 90 degree above, it have very little or no activity. So if you want to stop their activity, just rise the temperature or decrease the temperature. But rise in temperature is stopping their activity, but do not denaturing them, means do not degrading the enzyme, do not making enzyme non-functional. Enzyme is remaining intact, whatever if the temperature is 90 degrees Celsius or 97 degrees Celsius, depending upon their stability half-life. Okay, so then some more property of this enzyme that like as I mentioned, each enzyme has required some buffering condition, ionic condition. So different ionic condition are going to enhance the activity. Some ionic condition are there which can reduce the activity also. So KCl and magnesium ion, they promote the activity. So in the buffer, you must have the KCl and magnesium chloride so that the enzymatic activity is better. And TEC molecules have maximal activity at the 50 millimolar KCl. That is their optimal condition. So when you are using these enzymes, the buffer that you are going to make must have the 50 millimolar your KCl and just right concentration of MgCl2, which is determined by the concentration of nucleotide triphosphate. Similarly, you can also optimize your concentration of magnesium chloride and your requirement of your DNTPs. One drawback that your TEC polymerase is going to have, it's not the drawback that represents all the thermostable polymerases, but this drawback is there in your TEC polymerase. It lacks the proofreading activity, that is the 3', 5' exo nuclease activity is missing in your this polymerase. So it means if you use this tag polymerase enzyme for your DNA replication, then this activity is going to be error prone. Because if the wrong nucleotide is being incorporated, then it won't check whether it is wrong or right. However, the incorporation is not very frequent. There is only one wrong nucleotide among the 9,000 nucleotides it added. So you can rely on this polymerase, but when you want to show 100% that there is no error, in that case you can't use this tag polymerase. So for that, what you need an enzyme which must have this proofreading activity. So this proofreading activity is present in another thermostable polymerase enzyme that is called the Pfu Dna Polymerase. This Pfu Dna Polymerase is being isolated from again a hyperthermophilic archaebacteria, that is the Pyrococcus furious. So P stands for Pyrococcus, Fu stands for furious. As in case of tag, E stands for thermophilus, AQ stands for aquaticus. So these are the PFU. Wherever you required proofreading activity, you can't bear any error, you need PFU. In case you can bear the errors, you can use the tag polymerase. So simply, if you talk about the diagnosis purpose, in diagnosis we don't want to know the DNA sequence, so we can use tag polymerase. But for the sequencing, we can't bear the errors. We need PFU DNA polymerase. So there are some other polymerases also, which are thermostable, and as different sources are there. Amplitac low DNA polymerase we got from the Thermus flavus, TUB, TUB, that's the Thermus ubiquitous, then PWO, Pyrococcus vosai, then RTH, Thermus thermophilus, TFL, Thermophilus flavus, TLI, Thermophilus vitoralis, Ultima, Thermotoga martima. So these are different sorts, different enzymes. If you want to read about these enzymes more, you can refer this article, which is listed in the reference that is published in Applied Environmental Microbiology in 1998. So this is a bit about your thermostable enzymes. So anyone would like to ask about the thermostable enzyme, please let me know. So anything that you want to know about the DNA polymerase or the thermostable enzymes, how they act, what they need, how they work. Any query?
No, sir, no query.
Yes, sir.
Yes.
Sir, as you told that some of the enzymes are there, but then we provide a more computer, then they will not.
Very high background noise is coming. Are you in kitchen?
No, sir.
Okay. So please speak it again.
Sir, actually, I'm saying that as you told us that there are few enzymes which don't work when the temperature is high.
I'm not getting what you're saying. Shika?
Yes, sir. So,
Please
am
speak.
I audible?
Yes, now you're audible.
Sir, as you told that there are few enzymes, few thermophilic enzymes which don't work when the temperature is much high.
The temperature is?
When the temperature is much high, like the temperature they need, it is more than that.
Okay, yes.
Sir, what if suppose we provide temperature which is not good for them, but still they work, then
They will
that
work.
is possible.
They will work not like simply example as if I take example of you. So if today's environment is a pleasant environment, it is raining, but it is very humid. If anybody asks you to work outside and do some exercise yoga, then you feel that you will do, but you will not work properly. And similarly, humid conditions are not less humidity is there, temperature is very pleasant, then you will work very well. You can rush, you can run very long. So their activity is still there, but it will be less. You will be less active. So similarly, enzymes are also there, they are going to have activity, but their activity is going to be less. So activity less means if you see this slide, in this slide we mentioned that uh, sorry, yeah, this one. If you see that if your temperature is 72 degrees Celsius, it is adding 1000 base pair in 10 seconds. At immediate 75 to 80, it is adding 150 nucleotide per second. So further activity is high. If you're decreasing the temperature 70 degrees Celsius, its activity decreases to 60 nucleotides. Then for if you move to temperature 55, its activity is to 24. If further decreasing, its activity further decreasing at 22 degrees Celsius, just 0.25 nucleotide. Okay, so when you decrease, when you changing your optimal temperature, enzyme is still active. Enzyme can do the job, but it can't do properly. But it can be its full efficiency. If you want to work your enzyme simply as if you want to work with your full efficiency, you need your desired condition. If your desired conditions are not fulfilled, you are not going to work efficiently. Same thing is there for enzymes also.
Okay, sir.
Okay.
Thank you, sir. Yes, sir.
So this is the all about your thermostable polymerases. Now one more type of the polymerase is there that we call the reverse transcriptase or polymerase reverse transcriptase. So something here reverse is there. So reverse is not a way that we're talking about. The direction is reversing. If you say the DNA polymerase, then we are talking about they were replicating in the 5' to 3' of newly synthesized strand. And then the reverse polymerase, reverse transcriptase, they are going to synthesize in the 3' to 5'. No. So here the term is transcriptase. So transcript is there, transcription is there. So what is the transcription? What do you mean by the transcription? So what do you mean by the transcription?
Sir, it is the process.
Yes, Bolo Devanshi?
Sir, it is the process by which in a strand of a DNA, it can be copied into in a molecule of a messenger RNA.
Yes. So simply it is also replication, you can say, in which DNA to the formation of RNA is there. So over the DNA template, if RNA is forming, that step we call the transcription. So here we are doing the reverse through the transcription, means over the DNA template we are not making the RNA, but we are making DNA over a template of RNA. Okay, so in DNA replication always occur DNA replicate in the form of DNA. DNA also transcribe in the form of RNA. RNA translate into the proteins. But here we are talking a reverse thing. Reverse means RNA is replicating in a form, or means it's a translating not in the form of a protein, but it is a translating in the form of a DNA, or it is a replicating in the form of DNA. So that job is called as a reverse transcription. And the job of that reverse transcription, it means on the template of your RNA or a template of RNA, we are making DNA. So that job is done by another type of polymerase. So polymerase means a polymerization, and polymerization your DNA polymerase, that we call the reverse transcriptase. So reverse transcriptase is also type of a DNA polymerase. So how these two types of DNA polymerase are going to be differing? So now we can incorporate one more term in when we are describing this polymerase. One is called the DNA dependent DNA polymerase. This is a polymerase which depends upon DNA and polymerizing what? Polymerizing DNA only. So that type of called the DNA dependent DNA polymerase. And another type of polymerase that is called the DNA dependent RNA polymerase. Means an enzyme which is dependent upon DNA, which is going to use a DNA template strand but will synthesize a RNA. That is called a DNA dependent RNA polymerase. Now we are talking another type of enzyme. What it is going to do? It is going to utilizing RNA as a template, and it is synthesizing DNA over it. to those enzymes we called RNA dependent DNA polymerases. Okay, so I think our time is rushing out, so I'm stopping it here, and we will continue reverse transcriptase or reverse transcriptase in our next class. So before moving to next, we are having 5 minutes. This is a discussion session. If you want to ask anything, please let me know. Otherwise, we have to move for the attendance. Any query?
No, sir.
Okay, so when I recite your name, please turn on your camera. Demetrius,
Present, sir.
Tridev, Tridev, Manaswini,
Sir,
Present, sir.
excuse me, sir.
Hanji Tridev ji.
Sir, I'm present. My mic was not working.
Adish,
Present, sir.
Yuvraj,
Present, sir.
Sanjana,
Present, sir.
Nehal,
Present, sir.
Atoiba,
Present, sir.
Devanshi, Devanshi,
Present, sir.
Nandini, Nandini, Kshitij, Kshitij,
Present, sir.
Janvi,
Present, sir.
Shivani,
Present, sir.
Shakti,
Present, sir.
Rupal,
Present, sir.
Sahiba, Sahiba, Ishita, Ishita, Namrita,
Present, sir.
Present, sir.
Tanvi,
Present, sir.
Yukta,
Present, sir.
Ayushi,
Present, sir.
Vivek.
Present, sir.
Okay, so Nandini is absent. Rest all present. It is before ending. Who joined late today? Who joined late today? Or should I need to check your
So,
attendance sheet?
so I joined 5 minutes late because of my bad
No, I'm not
network. I
asking
was
5 minutes late. I'm asking who are 20-30 minutes late. Late means
no,
20-30.
not 30 minutes.
Late
It's
means
just 5
20-30. Late means
Okay, sir.
20-30.
Okay, sir.
Because one student joins, I think, around 20 to 25 minutes late. Okay, so I am excusing today. Next time I won't excuse. Clearly, I will mark them absent. First thing, you are not responding even after joining late. Okay, so stay safe, stay happy, enjoy the day.
Thank you, sir. Thank you, sir.
Thank you, sir.
Thank you, sir.